Your third and final video for chapter 3 is active transport. Active transport, you will remember, requires energy. And energy for our cells is ATP, adenosine triphosphate. We are going to have 3 different kinds of active transport: primary, secondary, and vesicular. And you're going to see there's multiple kinds of vesicular transport as well. There we go. So we are going to start. Oh, we're starting with vesicular transport? Okay, let's start with vesicular transport. So this is how you get large particles or a large amount of particles or fluid across the plasma membrane. So this is stuff that's way too big to get through a carrier because it's bigger than the carrier. And you basically use a bubble of the membrane. So it's kind of like when you scoop up your food with corn tortillas if you're having fajitas, or maybe naan if you're having a good curry dish. So it's pretty much the same way. The cell membrane just invaginates and forms a bubble and then pinches off at the surface, and whatever was in that little bubble that got pinched off is now drawn into the cell in a vesicle. So if you're pulling stuff into the cell, it's called endocytosis. There are 2 different types of endocytosis. If you're just sucking in liquid and whatever is dissolved in the liquid, it's called pinocytosis. If you are specifically picking things out of the solution by having them stick to surface proteins and then internalizing those surface proteins, that's called receptor-mediated endocytosis. So in this circumstance here, anything that's outside the cell gets drawn inside the cell. Over here, you're picking and choosing what gets drawn in. Phagocytosis is a special form of endocytosis. And in this case, you're internalizing very large solids. So something about the size of an entire bacterial cell, or maybe some cell debris, red blood cell that's fallen apart in circulation, or some cell that's been damaged because you got a cut or a bruise. So really big things that are sort of indiscriminately brought into the cell are brought in through phagocytosis, which phago means eating, and cytosis just refers to the cell, so this is cell eating. Any cell that is capable of performing phagocytosis is called a phagocyte, and then what you have here, macrophages, are just one example of a specific kind of phagocyte. Exocytosis is just phagocytosis in reverse, or endocytosis in reverse. So you have inside of the cell a bundle of usually proteins, something that you want to secrete out of the cell in a vesicle. That vesicle will be brought over to the plasma membrane, and then proteins embedded in the vesicular membrane will link with proteins in the plasma membrane, and that will essentially unzip the plasma membrane or the membrane of the vesicle, and then allow whatever was in the vesicle to be dumped outside the cell. So this is how your cells secrete proteins, neurotransmitters, and other polar molecules that they want to have control over when they go out. So they're not just passing through a carrier. Oh, I forgot. Here are some examples. Then endocytosis and transcytosis is basically endocytosis plus exocytosis. If you look at the figure in the book, they label all of these intermediate steps. All you need to know is that a cell has the capability of pulling something in on this side, transporting it across the cell, and then dumping it out on the other side. This is how certain things get from your digestive tract into your bloodstream. So think of a cell lining the inside of your intestine, absorbing or endocytosing amino acids on one side, transporting the amino acids across, and then dumping them on the other side of the cell where they can get taken up by a blood vessel. So the next kind of active transport is called primary active transport. This is pumps. Pumps are little proteins that burn ATP and shuttle stuff across the cell. So just like water, a sump pump can pump water out of your basement, or the fuel pump in your car can pump fuel towards the engine. Pumps in your cells can move or pump ions and other things into or out of the cell against their concentration gradient. So you're moving things from an area of low concentration to an area of high concentration, and that requires energy. It's like pushing a ball up a hill. The most prominent pump that we care about is the sodium-potassium ATPase. You can read this stuff over here. I'm going to talk about it on this slide here. So what the sodium-potassium ATPase does is pump sodium out of the cell and potassium into the cell. So here you have some sodium molecules which bind to the inside of the pump, 1, 2, 3, along with the phosphorus from an ATP, so there's some energy. That energizes the pump so that it can move sodium out of the cell against its concentration gradient. Then potassium binds to the inside of the pump, and the pump reshapes itself and pushes the potassium inside the cell. And the potassium is sort of lets go of the pump. Sorry, I'm stumbling over things. Lets go of the pump once it gets inside because the shape of the pump changes. And then it's ready to start another round of pumping sodium out and potassium in. So for every ATP this pump burns, it shuttles 1 sodium out of the cell, and 3 sodiums out of the cell and 2 potassium into the cell, both against their concentration gradient. Cotransport is a 2-step process. So in cotransport, it starts with a pump, and then the pump uses energy to generate a concentration gradient. That concentration gradient represents potential energy. It's like the ball or a ball at the top of a hill. So the energy that the pump puts into generating that gradient can then be used to move something else against its concentration gradient. And I'll show you a picture later that makes a little bit more sense of it. If you have a symport system, the ion and the thing that is also being moved move in the same direction, either both into the cell or both out of the cell. And then in an antiport system, the ion is being pumped in one direction and the other molecule is being pumped in the other direction or flows in the other direction. So here you have an example of a sodium symport mechanism or cotransport. So you have your sodium-potassium ATPase. It's pumping sodium out of the cell against its concentration gradient. So all of the energy from the ATP gets stored as this gradient of sodium, and then the cell can use the energy in the sodium concentration gradient to drive the transport of glucose. So you have this cotransporter that has a spot for sodium and it has a spot for glucose. And because there's a gradient for sodium, sodium can kind of force its way in and drag glucose along with it. So what you can do with this sort of process is move glucose against its concentration gradient by using some of the energy for the sodium gradient. So sodium is traveling down its concentration gradient, and the energy given up by that process of rolling sodium down the hill is used to drag glucose into the cell against its concentration gradient. That is active transport in 9 minutes.